Hello everyone, let us continue the study of recombinant DNA technology topic, and in this session we'll study the lambda phage vectors and the Cosmids, which are used as vectors in recombinant DNA technology. Lambda phase vectors. So what are phases? So bacteriophages are a type of virus which infect bacteria. So in one of my previous session, I told you about the viruses that infect bacteria. So they are called phage bacteria and they are commonly called bacteriophages. And structurally, viruses are very simple in structure. They consist of a capsule and the genetic material. So here structurally you can see the phage is made up of, the virus is made up of head, tail and tail fiber. So this is the head part and this is the tail part and tail fibers. So in this head, the genetic material of this particular phage is present. So the DNA is present in this head. The DNA which is found in the phage, the lambda phage, is a double-stranded and it is a circular DNA which is about 4.8 kilobase pair size. It infects Escherichia coli bacteria and during the infection only the DNA is injected into the bacteria. DNA produces multiple copies of phage DNA and it produces phages inside the host cell and bacterial cell bursts to give out new phages. So to begin with, you remember phage or bacteriophage is a virus that infects bacteria. So to understand more, let us see this picture. So this is a bacteria and this is the phage, the virus that is capable of infecting the bacteria. As I already told you, the DNA of this phage is present in the head region. So during the infection, what happens? You can see here, the phage will not enter into the bacterial cell. So it is going to insert only the genetic material, that is DNA, into the bacterial cytoplasm through the tail. So the phage works like a syringe which is going to inject the DNA into the bacterial cytoplasm. So that is the character. And once the phage DNA, you remember this bacteria is having its own DNA, but the DNA of the phage, once it enters into the bacteria, the phage DNA has the genes for the synthesis of head, tail fibers, etc., the particles. So all the genes which are required for the synthesis of phage particles are present in this DNA. But for the synthesis of phage particles, it is going to use the resources of the bacteria. That is the thing. So DNA is phage DNA, but the nucleotides, the proteins, enzymes, etc., it is going to use from the bacterial cell. So before that, there is a DNA replication. So just assume one phage infects, one DNA molecule is inserted, and once inside the bacteria, it is going to multiply. The DNA is going to replicate into many copies. Once multiple copies of DNA are formed, it is going to synthesize the capsule, the tail, the parts of the phage are going to be synthesized. And then there is assembly. The DNA molecules will become part of the head. So they are going to be assembled like phages. And once large number of phages are formed, they are going to burst out of the bacterial cell. So here you can see the host DNA, the host bacterial cell is going to be destroyed. So this is one cycle which is commonly called lytic cycle, where the infection occurs, the DNA copies are formed, the DNA produces the phage particles are assembled and the cell is going to burst out. So this is how the bacterial cell is going to be infected with phage. So in the genetic engineering, in the recombinant DNA technology, this property, so the ability of the phage to transfer DNA into the bacterial cell, so this property is important for us because our aim is to insert the foreign DNA into the bacteria. So this phage is capable of delivering DNA into the bacterial cell. So what we have to do is we have to insert the gene of our interest in the phage DNA, right? So once we insert that gene into the phage DNA, so the phage itself is going to deliver that gene into the bacterial cell. So in this way, the phage DNA can be used as a vector, as a tool of transfer of the desired gene into the bacterial cell. So lambda phage vector, some properties, let us see. Large DNA insert. So what is the difference between a normal plasmid and a lambda phage vector? Lambda phage vectors can carry relatively large DNA inserts of up to 25 kilobase pairs. So this is one advantage. Using lambda, we can insert large sized genes compared to the plasmids, which is larger than the most plasmids. This allows for the cloning of larger genes or DNA fragments. Site-specific integration. Lambda phage vectors can integrate into the host genome at a specific location called the ATTB site. This ensures stable integration and expression of the cloned gene. So this phage has a specific site called ATTB site, and the gene of our interest is going to be inserted exactly at this particular site, right? So this specific location is very helpful. It's of an added advantage because the integration is stable and expression is also stable. High transfection efficiency. Lambda phage vectors can be easily transfected into bacterial cells using standard techniques, resulting in a high transfection efficiency. So transfer of the recombinant DNA into the bacterial cell is more efficient when we use lambda phage. Dual modes of replication. Lambda phage vectors can replicate either as a plasmid or integrate into the host genome. This provides flexibility in the choice of cloning strategy and the ability to produce either high copy number plasmids or stable low copy number integrated clones. So I will explain this in a next slide. Non-pathogenic. So lambda phage is a non-pathogenic virus that infects only bacteria, making it safe to use in the laboratory. So when we use different type of viruses as vector, it may be very dangerous if they are pathogenic, but the lambda phage is non-pathogenic to human beings. So therefore, using lambda phage virus in a laboratory condition is safer. And lambda phage vectors can be easily detected by plaque assays, which allows for the rapid screening and identification of clones containing the desired insert. So as I already told you, once the recombinant DNA is inserted into the host cell, so there is a screening whether the recombinant DNA has entered into the cell or not, that has to be identified. And if we use lambda phage as vector, this process becomes very easy. And here is the structure of lambda phage. The main characteristic feature of lambda phage is the COS site, cohesive sticky ends, right? So we already know what is the sticky end. So sticky ends are created by the restriction endonuclease cleavage, right? And sticky ends help in joining together. So for example, these two are the sticky ends which can join with another DNA. So presence of this US site is the most important character of phage DNA or phage vector. Phage, bacteriophage, which is a virus, in a natural condition, it shows two types of infections. One is lytic cycle, another one is lysogenic cycle. So let us understand what is the difference between the lytic cycle of a phage and the lysogenic cycle of a phage. So in the lytic cycle, the phage infects the bacteria. So this is the bacterial DNA and this is the phage DNA, right? So the DNA is inserted into the bacteria and phage DNA becomes part of the bacterial cytoplasm. There is DNA multiplication, DNA replication, and this DNA is going to produce the particles of the phage, and there is assembly of phages. So one DNA is inserted here, multiple copies of DNA are formed, multiple copies of phages are formed using the resources of the bacteria. And at the end, the bacterial cell is going to break. There is a lysis of the bacterial cell and large number of phages emerge out. So one phage has entered, I mean one DNA, and it has resulted in the emergence of multiple phages. So each phage is capable of infecting one bacteria each. Since at the end of this cycle there is a lysis of the bacteria cell, therefore it is called lytic cycle. Now what is the advantage as far as genetic engineering point or recombinant DNA technology is considered? Here only one DNA is inserted and at the end we can get multiple copies of the DNA. So this method, this type of cycle is going to achieve the cloning of DNA, the cloning of genes. So we can get multiple copies of genes. So this is the advantage of lytic cycle. Whereas in natural condition, the same phage shows another cycle called lysogenic cycle, which is very interesting. The DNA molecule is inserted, and here because of the presence of COS, cohesive sticky ends, it will integrate itself into the bacterial genome. So you can see here, this is the bacterial genome. And the phage DNA, instead of multiplying separately, it has become part of the DNA of bacteria. So now it is part of the DNA chromosome. Now it is not going to form multiple copies of phage, etc. here. It is just going to be part of the chromosome. It is not going to produce the phages. Then what happens? When this bacteria multiplies, so here you can see there is a multiplication of bacteria. When this bacteria divides and forms two bacteria, there is multiplication of bacteria. Here there is one copy, and with increase in the number of bacteria there are two copies, right? So what is happening here is, with the multiplication of bacteria, the phage DNA is also multiplying. Since it is latent, it is not destroying the cell, we call it as lysogenic cycle, right? So the phages are not being formed, and after many cycles, say just imagine one bacteria is infected and multiplication continues. When thousands of bacteria are formed, each bacteria is having the phage DNA. When the conditions are suitable, this DNA can separate from the bacterial genome, bacterial chromosome, and it can show the lytic cycle here. So the DNA in each cell, here what happened, thousands of bacteria are formed, and in each bacteria the phage DNA can come out into the cytoplasm. It can start producing the phage particles and it can enter into the lytic cycle. So This is where we can get uh, the product, gene product. So this cycle, the lysogenic cycle, is very helpful in the recombinant DNA technology to get the gene product. Here we get multiple copies of the gene, but there are no gene products. But here the genes are going to be expressed and we get the gene product. So in this way, phage DNA is very useful as a gene vector. So the same thing I have explained here. Bacteriophage, the lytic cycle, uh, I already explained it, so there is no need to repeat, I think. And this is the lysogenic cycle, right? So that's about the lysogenic cycle and the lytic cycle of the lambda phage. So lambda phage is a suitable vector because uh, cloned phage DNA is packed in a phage particle, infection of host bacteria, and screening. So I already told you about this. So the DNA of our interest can be cloned in the lambda phage, right? So this is, okay, see here, this is lambda uh, phage DNA, and in this lambda phage DNA we are going to insert the gene of our interest, right? So here you can see, if this is lambda phage DNA, before inserting you can insert the gene of your interest, right? So like this it can be used as a vector. And applications, gene expression studies, recombinant protein production, and gene library creation. So these are the applications of using lambda phage as a vector. Next, another type of gene vector which are being used are cosmids. So cosmids are engineered vectors that combine characteristics of both plasmid and phage. You can consider it as a hybrid between a plasmid and a phage. It is formed by joining ends of a linearized plasmid DNA with COS sites, cohesive sticky uh, end site of lambda DNA. It is a derived vector, right? So you can consider it as a combination of plasmid and phage. It is a hybrid, right? It is produced by joining both plasmid and phage vectors COS site. The cosmid DNA can be packed in a capsid of lambda phage in vitro to form recombinant phage particles. So, what is the specialty of cosmid? So, this is the plasmid, right? And I mean a cosmid which has COS sites, right? We already know what are the parts of a plasmid. And from the phage DNA, they have taken these COS, cohesive sticky ends, right? And using the restriction endonuclease, the gene of our interest can be inserted in the restriction sites. So this is a DNA of, I mean the gene of interest. It is inserted in the cosmid. So this is a cosmid which is engineered. Uh, it is same. The technique is same as with the plasmid or phage DNA. But here, in case of plasmid, it is directly made to taken up by the bacteria, whereas phage is the one which will infect the bacteria. So what we are doing here is we are taking the plasmid and we are adding a property of phage, and that is a cosmid. So cosmid is a plasmid with COS site. You can consider it like that. It is a plasmid with COS site from the phage. This is going to be packaged into a phage, into a bacteriophage. So we are using bacteriophage to insert our cosmid into the host cell. I mean the E. coli bacteria. So this is going to transfer. So what is the difference? In case of phage, it is going to insert the phage DNA, but here it is going to insert the cosmate, which is artificially created and which is bundled into the phage. So that is the advantage of, that is the cosmid being used as a vector. They have been constructed to include fragment of phage that includes the COS site. Cloning into cosmid is similar to cloning in phage. I already told you there is no difference as far as the technique is concerned. It involves digesting exogenous DNA with restriction enzyme, cutting the cosmid vector with a compatible restriction enzyme, combining the two, and ligating them. Once the exogenous DNA is inserted into the cosmids, cosmids are packaged in a manner similar to that used with phage. Packaging the cosmid recombinants into phage coats provides a useful method for selecting the size of the inserted DNA. So, if the same thing can be achieved by using a phage, lambda phage, then what is the need for creating this cosmate? This is the difference. What is significant about cloning with cosmate is that larger DNA fragments, 32 to 47 kilobase, can be inserted into the vector and still be packaged. So this is the reason for creating cosmids. Of course, using plasmid, we can transfer gene into the bacteria. Using lambda phage also, we can transfer the gene into the bacteria. But the DNA, the gene which can be transferred, there is a limitation in those two cases. But here using a cosmate, using this hybrid, we can transfer a very large sized gene into the bacteria. So that is the main thing about the cosmates. After in vitro packaging, cosmates are used to infect suitable E. coli strain. Infection of E. coli involves injection and circularization of the cosmid DNA, but no phage protein is produced. That means it mainly uses the lysogenic cycle, not the lytic cycle. Transformed E. coli cells are identified on the basis of the resistance to a specific antibiotic. So we discussed about this antibiotic resistance sites while studying the plasmids. So advantages, the most important advantage I already told you is we can pack large gene into the cosmid and we can transfer it into the bacterial cell. The same thing can be can prove as a disadvantage, right? So the packaging enzyme fails to pack recombinant cosmid into a phage head if any one of the two co-ends is missing. While preparing the cosmid, if COS ends, so COS ends must be present on both the ends of the cosmid. So if it's not there, the process will fail. And sometimes more than one recombinant cosmid join together to form a large DNA. So this will again fail the procedure. Slower replication rate, higher frequency of recombination inside bacterial host, and unstable inside E. coli host and thus easy to lose vector. So these are some of the advantages of cosmids. Uh, so in this video we studied the lambda phage and cosmids. So for further studies and more reading, you can refer these reference material or you can refer the internet or the textbooks. So thank you.